Sulfonamide drugs were the first antimicrobial drugs that paved the way for the revolution in medicine. The first sulfonamide, trade-named Prontosil, was a prodrug, which is a substance that is administered in an inactive form and is then converted to an active agent through normal metabolic processes or bioactivation. Experiments with the new drug, Prontosil, began in 1932 in the Bayer laboratories, which were a component of a large German chemical trust. The Bayer team believed that coal tar dyes, which had the ability to bind to bacteria and parasites, would be useful in targeting harmful organisms within the body. After many attempts of dye testing that led to no answers, a team led by Gerard Domag finally found a dye that worked. It was a red dye that was synthesized by Bayer chemist Joseph Clare. This dye had remarkable effects on stopping some bacterial infections in mice. This dye was related to the azo dyes, which are characterized by their structure, which contains a diazo group. Their discovery was not published until 1935, more than 2 years after the drug had been patented by Clare and his research partner Fritz Maisch. Prontosil was the first medicine ever that could effectively treat a range of bacterial infections within the body. However, the drug had no effect while in a test tube, which led them to believe it had antibacterial effects only on living beings.
Using that bug to be killed in a test tube with this antibiotic and doing it in the human body, when they first came out with sulfa drugs, they put it in a test tube and it didn't kill bacteria. It wasn't until it passed through the body that they saw, oh my gosh, this kills bacteria. So it really created this concept of what you see in the test tube isn't necessarily what happens in the human body. And that created a whole line of innovations behind it, like penicillins and all this. So it was really the first drug to be tested and found to be antibacterial. Yeah.
Later, a French research team led by Fournet at the Pasteur Institute discovered that the drug was metabolized into 2 pieces within the body. This process released from the inactive dye portion into a smaller, colorless, and active compound, sulfanilamide. The discovery of sulfanilamide helped to establish the concept of bioactivation, which is the process of metabolic pathways activating an inactive form of a drug. The discovery also ruined the dreams of enormous profits because this active ingredient had been synthesized first in 1906 and was widely used in the dye-making industry. The sulfa drug was now available for anyone to make. The result was a sulfa frenzy. Hundreds of manufacturers in the 1930s were producing tens of thousands of tons of sulfa drugs. Testing requirements were non-existent in that day and age, leading to the elixir sulfanilamide disaster in the fall of 1937. At least 100 people were poisoned with diethylene glycol. This led to the passage of the Federal Food, Drug, and Cosmetic Act, or FDA, in 1938. As the first and only available antibiotic for many years before penicillin, the sulfa drugs continued to thrive through the early years of World War II. Many thousands of molecules containing sulfanilamide structure have been created since its discovery, approximately 5,400 different variations. Many have improved formulations with greater effectiveness and less toxicity. These drugs have a wide range of uses within the body, starting at acne to urinary tract infections. This antibacterial acts as a competitive inhibitor of the enzyme dihydropytorate synthase, or DHPS, an enzyme that is involved in folate synthesis. As an inhibitor, this microorganism will be starved of folate and die.
During the normal enzyme catalytic cycle, the substrate encounters an enzyme with a specific active site to which it binds, forming an enzyme-substrate complex. The enzyme then facilitates the breakdown of the substrate to its products, which part from the enzyme, leaving the active site free to catalyze another substrate as the cycle begins again. Competitive inhibition occurs when an enzyme encounters a blocker which mimics the properties of the substrate and binds to the enzyme's active site. Thus, when the substrate is encountered, the active site is not available for attachment and no reaction will occur. Non-competitive enzyme inhibition involves the binding of a blocker to the enzyme away from the active site. This binding causes a conformational change in the enzyme, altering the shape of the active site, which prevents the substrate from binding. No reaction will occur as long as the non-competitive blocker is bound to the enzyme.
Folate is a water-soluble B vitamin that occurs naturally in food. It helps to produce and maintain cells and is especially important during periods of rapid cell division and growth, such as infancy and pregnancy. It is needed to make DNA and RNA, the building blocks of cells, and helps to prevent changes in DNA that may lead to cancer. DHPS catalyzes a reaction between DHPP and PABA, or p-aminobenzoate, to form folate. PABA, 4-aminobenzoic acid, is an organic compound with a white-gray crystalline structure that is partially soluble in water. PABA is an intermediate in the bacterial synthesis of folate. 4-aminobenzoic acid is often referred to as vitamin BX. Humans lack the enzymes to convert PABA into folate, which is why DHPS is necessary to catalyze the reaction between DHPP and PABA to produce folate at a rate that is suitable for life. In more basic terms, the bacteria that invade your body that make you sick require folate, just like human cells do, because folate is required for cells to grow and reproduce. Humans are able to get folate through their diet. This is important because the sulfa drugs would otherwise harm our normal metabolic pathways. Since bacteria isn't able to take this nutrient in through food, the sulfa drug is able to inhibit their metabolic pathways to stop the reaction between PABA and DHPP, which means that folate is not produced and the microorganisms will not be able to reproduce, ultimately killing off the bacteria. Sulfa drugs are still used today to treat numerous bacterial infections and are constantly being tested due to growing bacterial resistance.